Thank you very much, Nikki, for those kind words. Um, Ladies and gentlemen, distinguished guests, good morning. My name is Anita Ghansah from the Noguchi Memorial Institute for Medical Research of the University of Ghana. And I am greatly honored to be doing this commencing keynote speech for the first Public Alliance for Genomic Epidemiology Conference. And for this, I'm so grateful to the organizers for the opportunity. In an era where information is expanding so quickly and innovation is driving so much change, I think that we need to change our horizon. We need to start thinking about some of our previously conceived ideas to make change, to make ready to accommodate change. And so we have to come together and see that there is a brighter future ahead. And I believe that the theme for this conference, developing collaborative bioinformatics for public health and beyond, sets that tone. And therefore, I am very privileged to be contributing to this dialogue. I'll start the journey to this 3 days by looking at what the challenges and opportunities we have with bioinformatics for public health. We will look at the themes that this conference presents and how they are going to help us improve on our various disciplines, bearing in mind that our idea is to incorporate bioinformatics into public health. Now, I'll start the journey by taking you to 1943. I want to explain to you how I think malaria is a delayed case for public health genomics. Now, this 1943 production by Walt Disney entitled The Winged Scourge, it's a production that describes how malaria was controlled in 1943. And the Anopheles mosquito was described as enemy number one, and you can see that malaria was a challenge, a major public health challenge across the globe. Now, it may interest you to know that the Office of Malaria Control in war areas was renamed the Centers for Disease Control in 1947. That's the US CDC. So you see how important malaria was at the time. Now, some of the measures they used to control malaria in the 1940s were basically targeting the breeding sites of the mosquitoes, so cutting weeds, dusting with insecticides, screening of windows, and indoor spraying, draining of stagnant water, and targeting the larvae by spraying with oil, and they also applied treated bed nets. These approaches are no different from what we are doing today. The only additions are that our new antimalarials, the artemisinin combination therapy, are different from what was used then. And we also have two vaccines, the RTSS vaccine and the R21 vaccine for malaria control. And these vaccines are yet to be rolled out across Africa. Now, in terms of disease prevalence, we see how Plasmodium falciparum, the most deadly parasite, is targeting sub-Saharan Africa, and Plasmodium vivax more isolated in the Southern America and parts of Southeast Asia. Well, over the years, we have seen improvement. We have seen change. Yes, malaria is still prevalent, mainly in sub-Saharan Africa, and it is a major public health concern. But there have been efforts to still reduce the disease burden, and thanks to the Bill and Melinda Gates Foundation and its collaboration with WHO to ensure that malaria is eliminated. That is the call they made in 2010. We have seen that about 2 million malaria cases and 11.7 million deaths have been averted as a result of numerous interventions that have been put in place. If you look at the plot, you see that the, all right, can you see? Well, you see that we started in 2000 with about 222 million cases of malaria, and this increased around up to 2003, where chloroquine was changed to artemisinin combination as a result of increased drug resistance. So as time went on with the new drug policy, we saw a reduction, and this virtually continued. We had a downward trend in disease prevalence until somewhere in 2010, when the Gates Foundation, in collaboration with WHO, decided that we need to eliminate malaria. And this was followed by an avalanche of increased interventions like the use of bed nets, indoor residual spraying, more testing and treating to ensure that we are treating malaria. And this saw a marked reduction in disease. But somewhere around 2018, 2019, we saw a stalling in these figures. So the question is, why has this reduction stalled? Now, the last statistics indicates that we had 247 million cases of malaria globally, of which the WHO Africa region contributed about 95%, and 619 deaths due to malaria, 80% of which were resulting from children under the ages of 5 in sub-Saharan Africa. And COVID-19 also contributed a bit to this disruption. Between 2020 and 2021, 663,000 more malaria deaths were recorded as a result of lack of access to these interventions due to COVID-19, and there were 13 million more malaria cases. But the question is, why such high figures, even forgetting about the impact of COVID? We know that the malaria parasite is the strongest evolutionary force to the human genome in our recent history. This is how our forefathers changed from hunter-gathering to agricultural use, and they were infected by the mosquitoes, and therefore we adapted by selecting for the sickle cell mutation. Now, so malaria is a very strong force, that we are aware. But with these interventions, we know that the mosquitoes have developed resistance to the anti-malaria, to the insecticides, and the malaria parasites have also developed resistance to the drugs. Artemisinin resistance has emerged in Southeast Asia and has spread, and now we have observed resistance to artemisinin in East Africa, and it is spreading. So this is a natural response of the mosquito and the parasite. So we need to monitor drug resistance. We need to monitor insecticide resistance. And it's not that simple to just monitor these two, because what happens is that the residual parasites after these interventions need to be studied. We need to understand how the remaining parasites will adapt to the environment after such interventions, that is drastically reducing the mosquito population. So we need to go into the genetic dynamics to study the evolution of the remaining parasites. And this is where genomics will play a role, and this is how we can develop more impactful interventions based on our deep understanding of the evolution of the parasites. Now, There's a need to embrace all these powerful tools driven by sequencing and computational methods that I know most of you have the capacity to build. This is how we can study genome variations and evolution in greater detail. If we do that, we will be able to come up with more actionable knowledge for strategic planning of control interventions. And we need to continuously monitor these strategically planned interventions to ensure that we are obtaining maximum impact. And then we must monitor to the point of raising alerts if something goes wrong. Now, let's look at some of the general uses of genomics or bioinformatics in public health. We know that pathogen sequencing is a way of showing the blueprint of pathogens to understand its characteristics, such as its diversity and the rate of mutation, or how much of a force of mutation a region within a genome would be. Now, in terms of transmission dynamics, if we look at sequences between two individuals or two groups of populations, we will be able to establish the source of infection and how it is spreading. For outbreak investigations, sequencing is the means of studying whether there's a single origin of this outbreak or multiple origins, and also to track variations in these pathogens. Now, for drug resistance, our knowledge of a sequence of a pathogen that is resistant to a drug is important to help make decisions on the use of drugs or changes in drug policy. Now, genomics can also be applied in One Health, because if there is a zoonotic disease, studying the pathogen in both the animal and the host, the human host, will help us understand the biology of the disease. Now, in terms of vaccine development, understanding the diversity of the vaccine antigen will help us choose highly conserved regions within these antigens for vaccine design. And for surveillance and monitoring, I think that continuously monitoring these pathogens in a population is the way we can promptly avert any spread when it happens. And our phylogenetic trees will help us establish the evolutionary relationship between pathogens, look at new variants and the like. And all of these together will help us build public health policies based on results from these genomic studies. Now, a lot of diseases, both communicable and non-communicable, have taken so much advantage of the uses of genomics in public health. Rare diseases have been able to develop orphan drugs as a result of studying genomes. Cancer prevention and treatment, a lot of work has been done in cancer. Other diseases like kidney, chronic kidney diseases, hypertension, diabetes, have taken advantage of genomics to look for diagnostics, drugs, pharmacogenomics, and the likes. And genomic epidemiology in infectious disease control is taking deep roots for diseases like tuberculosis, HIV. Every TB genome can be sequenced, and they have panels of diagnostic tools ready as a result of studying the genomes, and so is HIV. Now, I know that precision medicine for some of these diseases is just a matter of time here in Africa, but we also know that COVID-19 has taught us a lot of lessons, or the coronavirus in general. This is not to bore you with this complex roadmap, but just to show you that advances in genomic epidemiology of the coronavirus has coincided with advances in sequencing technologies. So right from SARS-CoV in 2003, 2004, the genome of SARS-CoV was sequenced, and this became a reference for COVID-19 or SARS-CoV-2. H1N1 was sequenced, MERS-CoV was sequenced, and a lot of tools have been developed based on the sequencing technologies available. For COVID-19, we have had the RT-PCR assays quickly developed by January 2020, and then we had the Johns Hopkins University dashboard released in 2020, January. We had the COVID symptom app ready by March 2020, and by May 2021, we had the real-time SARS-CoV phylogenetics launched. That is the coronavirus. And COVID-19 itself has produced an extensive public awareness. The public, based on their own diagnosis, understand some level of genomics. We also have labs equipped with sequencing technology, thanks to Africa CDC's Pathogen Genomics Institute. And we have extensive training. Scientists in Africa, technicians have been trained to analyze our own data in Africa, all through the efforts of Africa CDC's PGI. But we know that unlike COVID-19, we cannot compare the scale and urgency of the global approach towards a complex pathogen like malaria, obviously because malaria is not a pandemic and it is mainly secluded to sub-Saharan Africa. So the urgency is different, but there is a lot we can learn from COVID-19. But we are not seeing this with malaria. This is why I think malaria is a delayed case of public health bioinformatics or genomics compared to other diseases like TB, HIV, diabetes, and the likes that I've talked about. So why is it so? The life cycle, the biology of the Plasmodium falciparum is very complex. It is an interaction between three genomes: the malaria parasite itself, the vector genome, that's the mosquito genome, and the human genome. So the malaria parasite has the duty to combat various phases in the vector. And after crossing this barrier, it has to enter into the bloodstream to the liver of the human. And it has to overcome immune responses over there, then reaches the red blood cell of the host. And within the red blood cell, that is where it actually propagates. It needs to overcome a lot of immune responses in order to survive. Now, an infection can be either monoclonal or polyclonal. That is also another level of complexity that we have to deal with when you are sequencing or dealing with the malaria parasite's genomics. Now, there is a very high mutation rate, and the malaria parasite is able to overcome all these by conducting extensive genetic diversity. It has to change at every stage in order to survive in these three genomes. And so this makes the data generation very complex, the data analysis very complex, and by the way, the genome itself is very big compared to genomes like bacteria genomes, viral genomes. So it's very big, and it is about 80% AT rich, which makes sequencing very difficult. So you see all these complexities add to the reason why I say malaria is a delayed case for pathogen genomics for public health. And it makes the cost also very expensive. Now, It is not all that gloomy. It's not all gloomy. We have made a lot of progress with malaria genomics, and I will show you some of the examples of how we have applied genomics for malaria control in Ghana. We have done this in 4 different areas: drug resistance monitoring, diagnostic resistance monitoring, vaccine efficacy studies, and insecticide resistance monitoring. Now, since 2003, the Noguchi Memorial Institute, in collaboration with the National Malaria Control Program in Ghana, have applied the WHO recommended approaches to look at therapeutic efficacy of antimalarials, and these utilize very traditional methods of amplifying genes that have been associated with drug resistance and then digesting to genotype to see whether we have resistance or not. Lately, you add maybe Sanger sequencing if you have the capability, and this is done now every 2 years. So we have tried to incorporate some of the new deep sequencing approaches to look at malaria drug resistance in Ghana, and we have fed our data to the control program. This is an example of using the molecular inversion probe approach to target genes of interest, specifically drug-resistant genes. And the advantage of the MIP, or the molecular inversion probe, is that it is highly multiplexable because you are targeting different genomes or different regions within the genome of the malaria parasite. And we have used this to look at drug resistance in the sentinel sites, 2 sentinel sites in Ghana. And the results of this study not only provide the needed data, but we've also, because of the sensitivity of these deep sequencing approaches, found new mutations that need following or monitoring to see how they are going to impact the drugs. Another thing that we have done is whole genome sequencing to look at vaccine antigens. This study was conducted by sequencing the whole genome of the malaria parasite, but focusing on the circumsporozoid protein, which is the antigen that was used to formulate the 2 malaria vaccines that are yet to be fully rolled out in Ghana. And our results are stunning. There is extensive diversity in the antigen across 2 different ecological regions in Ghana. The red region is a region of high seasonal malaria in Ghana. And then the blue region is a region of low seasonal malaria or perennial malaria, and this is along the coast. And we see that the 3D7 strain that was used to formulate the vaccine is about 70% in the red region and only just 10% in the blue region. And this has a very important implication for the efficacy of the vaccine in just 2 districts or regions in Ghana. And such studies, we think, must be continued as the vaccine is rolled out in the country. Now, we are also aware of the cost of doing some of these next-generation sequencing. So we are coming up with innovations to try to reduce the cost of deep sequencing. We are involving the staff who are based at the district to collect samples that we're going to use for genomics. Previously, we would pitch camp as researchers, go and stay in the field for, say, 2 months to collect 100 samples for drug resistance surveillance. But now we have trained them to collect our samples. That is cutting down costs. And then when these samples arrive in our labs, what we do is to pool them, just as what was done for COVID. A pool of 10 samples means reduction in the cost by 10 times. So we pool these samples, and we quickly prepare our libraries, and then we do the sequencing, and we look at the frequency of these markers of resistance. And this has shown to be highly concordant, about 99.5% concordant with sequencing individual samples. And therefore, we have rolled this out in 10 sites across the country. It is our dream that this will become a hospital-based thing. Every hospital will drop blood into a tube and they'll be used to quickly screen for anti-malaria resistance. And if need be, we go to specific sites where we have seen signs of resistance. This way we think will reduce the cost drastically of doing genomic surveillance. And also, when we reach malaria elimination, where it's going to be very difficult to find individual samples, we believe that this pooling and screening will be the way forward. Now, we've also used sequencing approaches to look at complexity of infection when an intervention is going to be put in place. So the plot below shows us how complexity of infection has reduced from 3 pre-IRS intervention, that's the pre-spraying at 3 to 1.5 post-IRS interventions. And this is important for the control program to see the impact of some of the interventions they are putting in place. Now, we also look for diagnostic resistance. The rapid diagnostic testing that is done to diagnose malaria targets mainly the histidine-rich protein 1 and 2. And there have been reports of deletions in this histidine-rich protein. As a result, we may have individuals testing negative when in fact it is due to a deletion in the histidine-rich protein of Plasmodium falciparum. We can have cases where we have false positives as a result of residual histidine-rich proteins, which can circulate in the blood even up to a month post-infection. So you can pick those as positives. And also, if the infection is not just Plasmodium falciparum, but another species, let's say Plasmodium malariae, you could miss that target and classify the individual as negative, when in fact the person has got another form of malaria. So at Noguchi, we sequence individual samples to ascertain the level of histidine-rich program, and this is part of the malaria controls activity mandated by WHO. We are also developing dashboards so that results from, say, monitoring of drug-resistant markers can be made easily accessible and easily interpretable to the malaria control program. This is something that we are still working on. Our idea is to present dashboards just like what was done for COVID-19 so that we will see what is happening in the country in terms of malaria interventions and drug-resistant, diagnostic resistance in real time. Now, yes, we have done something, but we face challenges, and there are recommendations on how to deal with these challenges. Most of the data you have seen are results generated by scientists working in silos in their labs. So there are many people in Ghana doing different things, and we all feed our results to the malaria elimination program, and they are interested in seeing what the results look like. That is not the approach. What we need is to build a holistic ecosystem to effectively do this. We need to customize our standards and processes. We need harmonized processes. At least I know that in terms of sample collection, we can come up with a simple, a highly harmonized process. We need equitable and beneficial national policies that will help with data sharing. There are a lot of challenges. We need equitable data sharing. Sometimes, because of more complex analysis, your data falls into another hand, and then it becomes who owns this data. You know, there are major challenges with data sharing, and we need equitable data sharing. Now, thanks to Africa CDC, Sub-Saharan Africa has now been equipped with sequences. People are able to analyze data on the continent. But in terms of malaria, you find that there is limited data generation. The methods for generating the sequence data are still being optimized. And therefore, you will sometimes see that some of your samples may have to go to a collaborator. And so there is limited data generation on the continent, data management, and analysis. We can't ask for more bioinformatics capacity on the continent. We need a toolkit to analyze such complex data. We need user-friendly analytical pipelines. Biologists, I don't think, are interested in becoming coders, no. But we need the ecosystem that will provide us with user-friendly analytical pipelines so that we will analyze our data. So long and short-term capacity building is still needed on the continent. Now, ethics. Ethics is a major issue when it comes to genomics because our way of consenting, we have seen that even as scientists do not know what we mean by future studies, what kind of future studies we are going to conduct with genomics data. And so how do we consent? How do we obtain informed consent from a study participant? We need to look more in depth into the ethics of doing genomics. Now, effective communication of our scientific findings. How do we communicate our findings to policymakers, even amongst us scientists? How do we talk about our study findings, let alone get into the community? Now, at the community level, there is a need to engage these communities as partners and not just a means to obtain data. You'll be surprised to see that some of the people understand basic heredity. They understand why we look like each other or we do not look like each other in families. So engaging them as partners gives us the buy-in to conduct genomic studies. And of course, funding. We need more funding. Funding from our government, funding from our international donors is very important. But especially for the African government, we need their buy-in. They need to understand in their own way. We need to speak some kind of political language for them to see the importance of doing these studies, to get their buy-in, to put a bit of money into the budget for genomics. Now, all this work that we have done at Noguchi is a result of support from our former directors. Delgim as a training grant by the PDNA to train people in genomics in Africa. We succeeded in training a number of masters and PhDs in bioinformatics. Then the Ghana Health Service has been of great support as our partners. NIH has funded a lot of our work. Malaria Gen, where I was giving birth. And then our collaborators from Brown University. Currently, I am a Callisto Junior Science Fellow, thanks to the Bill and Melinda Gates Foundation, and this is also supporting part of the work we are doing. Now, to conclude, I'll say that we can incorporate bioinformatics into public health. But what we need is equitable partnership and collaborations. We need the support of an ecosystem to harmonize our processes. We need to build capacity in bioinformatics and genomics, in communication and data sharing, and even writing policies that our policymakers will read and incorporate. But as a malaria scientist, I would say that complex diseases need more attention. Now, we need to take advantage of emerging technologies, targeted interventions, In this era of predictive modeling and artificial intelligence, we need to incorporate all of these into developing bioinformatics tools for public health. And it is time to leverage on the lessons learned from COVID-19 for public health genomics. Otherwise, what will happen is that all the infrastructure and capacity that Africa CDC has put on the continent will come to waste. We need to leverage on these and scale up sequencing for complex diseases and any other discipline that one is working in. It's time to form genomic epi alliances, like what PHA4GE is doing, and to tackle complex diseases like malaria. Now, I want us all to remember that based on the statistics that I shared, 619 children, mainly in sub-Saharan Africa, died from malaria in 2021. This means that within the 60 minutes of my presentation, 60 children under 5 have died from malaria. We can contribute our quota to build this ecosystem of genomic epidemiology for public health if we work together. Ubuntu. Thank you.